Can you all hear me? Can you see me? I can hear myself quite well.
Yeah, we can hear you and see you and see your screen.
Excellent. I've been promoted to be panelist. I'm going to turn off my text messages. There's a bit of feedback, but I'll just ignore that. All right. Thanks very much. It's a pleasure to talk to you. I wish I could be there in person. It's kind of fun to be the second Canadian talking about RNA. I'm going to, being that I'm in the session about RNA, we'll be talking about whole transcriptome sequencing. I'll really focus on this section of my talk, the new methods to really diagnose childhood cancer. I'll show you lots of examples from our work in sarcoma, and we'd be thrilled to talk to you all virtually or at some other point about how we can apply this to the CBTN cohorts and brain tumors going forward. So why consider RNA when looking for creating an atlas of childhood cancer? Well, we know that genomic complexity, which is frequently found when we sequence whole genomes in the forms of chromothripsis, massive burdens of mutation, heterogeneous subclones. A lot of these things actually result in very clear transcriptional programs. A lot of those changes don't necessarily drive the tumor, and they can be sort of confounders when trying to find what actually does. The RNA, in contrast to DNA, is something that is quite dynamic. It represents the current state of the tumor, whereas the DNA and sometimes the methylation state represents a lot of the insults and the ancestral history of where the tumor came from. Motivated by this, really, if we created an RNA atlas, we could create quantifiable molecular assessments of childhood cancers. So I'll present a framework, and I'll just try and move things around here so I can see what I'm presenting. And so it's actually a two-part framework. The first is called Raccoon, both of which are the brainchild of Dr. Federico Comitani. The second bit on the bottom is called Otter. Raccoon is a method by which we subclassify in an unsupervised, recursive manner childhood tumors. Starting from a large data set, which I'll describe in a minute, we can remove a lot of the low information signals, reduce the dimensionality, and create these very informative clusters. The design of our method is such that it is, as I mentioned earlier, recursive, it's unsupervised, and it's scale adaptive, meaning that it's as good at creating small little leaves on a tree as it is at distinguishing these massive trunks or branches from one another. You get this really fine-grained detail, no matter how deep you zoom in, and I'll show you some examples of that as we go forward. In terms of the classifier, which we call Otter, its design was purposely done such that it doesn't rely on the labels of the tumors, so i.e. what the tumor was classified as, how it was signed out, and it uses the full transcriptome. What does it output? So for here, having run 13,000 tumors, we have 455 different classes and subclasses and subclasses of cancer, spanning the age ranges and organs in which cancers are found. Gives us this really detailed hierarchy of cancer, as well as normal tumors, I should say normal tissues. We included GTEx in here as well. What does Otter provide as output is very accurate probabilistic tumor assignments for individual patients, where you can say, for example, this is a breast cancer, this is the subtype of breast cancer, or this is the subtype. Before employing it, this tool, we tested its accuracy across a number of different measures. One, as I mentioned, this is a hierarchical approach. And you can see that the accuracy of the tool is still high, no matter if you're looking at the first hierarchy of cancer or if you're digging deeper and deeper into the 8th hierarchy. We then, in silico, added different tumor normal ratios, and you can see, for example, across multiple different tumor types, here's breast cancer, for example, kidney cancer and others. As you begin to add more and more normal, the major tumor assignment begins to flip. And depending on the strength of the tumor signal, that position where the flipping happens is different. We also tortured the algorithm in a number of different important ways where we removed portions of the transcriptome, creating essentially dropouts for certain genes. And as you would imagine, as you begin to remove portions of the full transcriptome, the accuracy begins to drop. But actually now we know how much signal we need for individual tumors to be able to accurately classify cancers. What's really cool is that there's a lot of room for improved efficiency and accuracy as we get more and more samples and more data. Most of the RNA sequence samples that we've looked at so far have been deeply sequenced, which means that they've been sequenced over 50 million reads, sometimes over 100 million reads. But when you subsample the tumors, that is, you selectively remove the reads, you still get massive high similarity, very high accuracy, even down to something like 5 million reads, which is a very low number of sequencing reads. It's actually difficult to get that little data off of a modern sequencing machine. We also tested the approach using multiple different algorithms for doing the alignment. For people who are sort of the RNA-seq mavens in the room, whether you use Callisto or STAR, whether you quantify the expression using a number of different ways, the accuracy stays quite good. And it really, in terms of running our classification, only takes about 5 minutes. And beautifully, where we are right now is, even having looked at 13,000 tumors, we're really at this linear phase where as we add more samples, we're getting more and more subtypes. So really, there's a lot of room for additional discovery as new subtypes are being discovered with additional tumors analyzed and added to this atlas. Okay, so we use this technology to study all of childhood cancer, but I'll just show you a few vignettes from mesenchymal cancers, from sarcomas, and show you what was used as input and what some of the interesting features of sarcomas are that were discovered using this approach. So as many of you know, this is a very complex area of oncology and of pathology, where there are over 60 malignant types of sarcoma and many benign entities as well. We used data from Target, TCGA, St. Jude's, and critically, the data that had been processed by the Treehouse Consortium, as well as some of our own data, and run it through this unified pipeline, including through Raccoon, to derive these 26 major types of cancer, spanning, again, the age range from pediatric to adults. In here, there are 26 main types, and you're looking at the topmost level. This is our hierarchical algorithm, so as you go deeper and deeper, you'll see finer granularity. For example, within breast, brain, or other places, you'll see subtypes and subtypes. For childhood cancers, they are quite interesting in that they are constrained to only 6 of these 26 main types, but they have remarkable deep subtyping, especially within brain cancers, going up to 8 levels deep within this hierarchy. In comparison to adult, primarily carcinomas and non-brain tumors that are really spread throughout this hierarchy and really tend to go maybe 2 or 3 different levels deep. Focusing on sarcoma, there are 2 major clusters that we already see at the main level. And actually their difference was surprising. We expected to see one massive mesodermal, mesenchymal cluster, but in fact we see 2. And the major difference is the degree of stemness. So looking at measures of stemness score, you see that there is a group of mesenchymal tumors of sarcomas that are called stem high. That includes rhabdomyosarcomas, both those that are driven by fusions and those without fusions, synovial sarcomas, Chick-Duck-4 tumors, and others. And then separate from that, quite distinctly, are the stem low tumors. And the stem low tumors, in fact, have high degrees of immune activity as measured by multiple different tools. And also, as you'd imagine, based on the names of the tumor types that I've shown you over here, this is primarily a class of childhood sarcomas, whereas these are adult tumors. We validated the stemness of the pediatric cohort by splaying a lot of the data onto a single cell fetal data set derived from the bone. And sure enough, you can see that those tumors, the bulk tumors that are high stemness, are completely indistinguishable almost from the single cells from the fetal bone. There's some really interesting outliers here, allowing us to study specific tumor types with greater detail. My lab has had a longstanding interest in Ewing sarcoma. And interestingly, Ewing seems to almost not be a sarcoma. It seems like a misnomer. It separates at the topmost level, separated away from all other bone and soft tissue cancers. It has no descendant tumors, meaning that you can't then subtype it any further. And this tool has really allowed us to fix a lot of the misdiagnoses that we've seen in Ewing sarcomas, where 14% of the tumors in this large data set that we've looked at were in fact tumors driven by CIC or B core rearrangements, usually fusions involving those two genes. So once you've created these hierarchies and these clusters, you can begin to ask questions about how much variability there is within a certain tumor cluster. So for this, Federico created a measure of transcriptional entropy, where, for example, you might have a tumor type over here in blue, where the genes are relatively consistent from sample to sample. So they're all kind of reddish, bluish, or mixed. And you have other tumor types or tumor clusters where there's lots of variability from sample to sample between individual genes. We aggregated the median of this variability in terms of these entropy measures to distinguish tumors. And again, we're looking now within clusters, within subtypes. And you can see that there are some tumors that have a specifically high degree of what we call transcriptional entropy. You could then ask yourself, well, which of the genes within that tumor type contribute to its entropy? And it turns out that the genes that are most important for the tumor's classification, so that is the disease-defining genes, are also those that are the most entropic, contributing disproportionately to the amount of entropy in individual tumors. So to just make that more succinct, the Ewing sarcoma, the genes that define Ewing sarcoma as distinct from the others are also those that are the most entropic compared to other cancers. So then we asked, how does this entropy change with age? And it turns out that pediatric cancers are much more entropic than adult cancers. And you don't have to really understand or go into great detail in terms of this graph on the left. Shows you the adult tumors over here in blue, the normal tissues over here in gray, and the pediatric tumors and their entropy on this x-axis shown on the far right. And what you can appreciate also, if you look at the graph, the violin plots on the right, or the half violin plots on the right, is that even when you look at leukemia versus leukemia, brain versus brain, and tumor type versus tumor type, within those entities, there is much higher entropy within pediatric cancer than there is in adult tumors. We're not sure whether this high degree of entropy is just related to the age of the patients or something to do with the cell of origin or preservation of a kind of a stem-like state in much of these tumors. But irrespective, what we're seeing is a lot more transcriptional possibilities within childhood cancers. And so what we are doing, as I showed you, that as you add more data, the definitions become more accurate. Josh Nash is a PhD student in my lab who's really been doubling the size of the sarcoma cohort by adding in different entities that we only had small numbers for before, and applying for data from around the world. So if we hit you up for a DPCAP application and you've granted us access, thank you so much. It's made a massive difference. And also what Josh is doing with help from the recent support from a XO1 that we received from Gabriella Miller as part of the CCDI is sequencing the paired genomes for a lot of these samples as well. So we can ask questions about the genomic underpinnings and the clinical consequences of these clusters that we're finding by RNA-seq. A lot of these samples that we're submitting to the XO1 come from, about within 500 meters or so of where I'm sitting, hospital across the street at Mount Sinai, or a biobank at SickKids, and they all have clinical annotations because they've been curated over a large number of years, primarily by Rebecca Gladdy, Irene Androulis, Jay Wonder, and David Malkin. And they're undergoing, importantly, a critical pathology review, really with a very modern lens to understand, to make sure that these entities haven't changed. So how are we using this tool going forward? We have an ongoing precision medicine program at SickKids where about 600 patients have been enrolled so far For these patients, we sequenced the whole genome, as well as a deep gene panel of 800 genes, and we also perform full RNA-seq. So we're using the OUTLIER framework to match individual ongoing patients to that atlas, and again, derive probabilistic class assignments. And as part of our tumor board meetings, we begin to stare at these plots where the wedges tell you the probability and directionality of assignment. In this case, kind of this probably is brain tumor. This shows you the subtype and then subtype with different degrees of p-values of those assignments. And importantly, critically, you can sometimes have the tumors matching to multiple different locations at the same time because it's multi-class. So here are two examples out of many. Here is, for example, an undifferentiated sarcoma on the left, which is rediagnosis of B-core associated sarcoma. And that small little wedge in kind of a dark blue-green represents the B-core subclass. After finding this RNA-seq based subtype, we reevaluated our cancer panel data, and sure enough, there was a small tandem duplication in B-core, which we missed, and we've since tweaked our algorithms to make sure that we find those going forward. But this is a great check of a parallel technology, and also showing you that the B-core alteration really manifests itself as a dramatic change to the transcriptional program. Similarly, on the right is a leukemia, which is diagnosed as a precursor B-ALL and re-diagnosed on the basis of the OUTLIER assignment as being pH-like. And here you can see that the probability extends almost to 100% of this subtype of pH-like leukemias. So overall, how well do we do? We were able to confirm or update the pathologist diagnosis for a good number of tumors. I'm seeing someone coming to the podium, but I can't hear them. I'll take their presence as a signal that I should hurry up. Good. So here we see that for the cases from Toronto, we'd be able to match or update the pathologist diagnosis for 89% of the cases. And with collaborators in Australia, we've been able to do the same thing for 95% of their cases. Overall, about 600 tumors have been used for this held out validation cohort. The other use of this technology is you can begin to use it to validate human models. And so, for example, we can quantify the Ewingsness of a number of different Ewing sarcoma models, both those with that have been unperturbed, as well as those where they've undergone CRISPR-Cas9 induced knockdown or deletions, and seeing how it reduces the Ewingsness of these tumors. We can do this, and we have done this for a number of different models. And finally, I just want to say that we've generated a early web portal that allows people to upload their RNA-seq for ongoing patients and then match it to the atlas. There are 3 neural networks that are running on a server at SickKids. And so if you're patient with this server, we'd be very happy to provide access and allow you to match ongoing patients, and thrilled to get your feedback. And so in summary, what I've tried to show you is that there's a novel computational pipeline that we've developed, allows you to understand transcriptional differences between and within sarcoma subtypes. It can be applied to other tumors as well. We see the major classes of mesenchymal tumors as being defined by age, stemness, and their fusions, of course. And this can be used as a diagnostic aid for ongoing patients, as well as to help us in terms of our functional assessment of in vitro models. We'll just end there, show you a picture of the team, and show you that there's a couple seats that are still empty. So if you're looking for an exciting postdoc position or PhD position, please get in touch.